Advanced IDs in this webinar, an open call entitled Accelerating AMR Diagnostics from Innovation to Clinical Impact. My name is Alec, together with Luca and Clevin. We are the project leads for Detect, and we are your moderators for today's session. So today we have a great lineup of speakers who will talk about how to improve rapid diagnostics use, especially in low resource settings. So this will be followed by the details of the open call in the second half of the session. So just a reminder that all questions will be entertained at the end of the sessions. So you may also type your questions in the chat box so our team can take note of them with the format on your screen. Without further ado, let me introduce our first speaker, Professor Ritu Banerjee. Professor Ritu is a professor and a pediatric infectious disease specialist at the Vanderbilt University Medical Center. Professor Ritu, over to you.
Hi, good evening, everybody. Let me just share my screen. Okay, can you confirm that you can see the screen,
Yes, Prof.
everybody?
You can go
Okay,
ahead.
great.
Thank you.
Okay, great. Okay, great. Pleasure to join you this evening to tell you about lessons learned from the FAST trial, which is a clinical outcome study that was recently published. It was funded by the Antibacterial Resistance Leadership Group, or ARLG, of the National Institutes of Health. So I have nothing to disclose. So by way of background, we all know that sepsis due to bloodstream infections has high mortality, up to 30% or greater with ineffective therapy. And the problem is that conventional blood culture evaluation is a lengthy process. Organism identification and susceptibility testing results are often available more than 2 days after a blood culture turns positive. And during that time, patients may be on ineffective antibiotic treatment, which leads to poor outcomes, or they may be receiving overtreatment or unnecessarily broad antibiotic treatment, which selects for more resistance and adverse events. So what we really need are faster tests that can shorten time to optimal therapy. Prior studies, though, evaluating blood culture diagnostics have had unclear clinical benefit, and often they were observational in nature or limited in power and not conducted in areas with high antibiotic resistance. So the FAST trial objective really was to compare clinical outcomes among patients with gram-negative bloodstream infections who have isolate antibiotic susceptibility testing determined using a rapid phenotypic method versus standard methods in areas with high antibiotic resistance rates. So here's the design of the study. We took patients who had blood cultures that demonstrated presence of gram-negative bacilli on gram stain. We randomized 850 subjects to either the rapid testing arm or the standard testing arm. This randomization happened in the laboratory. In the rapid testing arm, organism identification happened using MALDI-TOF. Then the rapid AST method was used. And then the standard AST method was used in addition, and antimicrobial stewardship review happened for all patients. In the standard testing arm, all the same thing happened, except there was no rapid AST test. And the rapid diagnostic that was tested in this trial was the Vitek Reveal, which is an approved diagnostic in Europe and the United States that measures changes in volatile organic compounds in the presence of bacterial growth. The turnaround time is fast, about 5 to 7 hours. And then we conducted the study in 4 countries highlighted on this map: Spain, Greece, Israel, and India. Okay, and the outcome, this was a clinical outcome study. So the primary outcome was a composite hierarchical clinical outcome called DOOR, or Desirability of Outcome Ranking, at 30 days. The worst outcome was alive with no deleterious events. That was the best outcome. The worst outcome was death, and the intermediate outcome was alive with 1 to 3 deleterious events. And you can see the events listed in the table below. And then there were a variety of secondary outcomes, which you can see: mortality, length of stay, time to antibiotic changes, etc. So here's the results of the primary outcome. There was no difference in door between the rapid testing arm and the standard testing arm. So the probability of the door being superior in the rapid testing arm was 48.8% with the confidence interval spanning 50%. So we concluded that the rapid testing arm was not superior to the standard testing arm. Here's another way to visualize that. You can see there were no differences between the arms in the proportion of patients who were alive with no events, alive with one event, or who had died at 30 days. And here are the results of the secondary and exploratory outcomes. In the top half of the slide, you can see mortality, length of stay, readmission, et cetera. There were no significant differences in the proportions or medians between the arms. However, when you look at antibiotic treatment and stewardship, you can see that the rapid testing arm had 14 hours median faster time to antibiotic escalation or de-escalation, and a greater proportion of subjects who had antibiotic stewardship recommendations made, and a greater proportion of subjects in whom stewardship recommendations were followed. And these were statistically significantly different between the arms. We also looked at a variety of subgroups. The antibiotic resistant subgroups actually had the most interesting differences between the arms. And I want to call your attention to, in both the cephalosporin resistant group and the carbapenem resistant group, there was a trend toward shorter length of stay in participants in the rapid arm compared to the standard arm, although these differences were not statistically significantly different. But then when you look at the carbapenem resistant arm, you could see that the proportion that were remaining in the hospital at 30 days was significantly less in the rapid testing arm compared to the standard testing arm. And then we looked at time to first antibiotic change, either escalation or de-escalation. And here I'm showing you time from randomization on the x-axis. And the standard arm is in blue and the rapid testing arm is in red. And you can see in all randomized patients, the rapid testing arm changes happened 14 hours faster compared to the standard arm. But as you got increasingly resistant infections, you can see that the difference between the arms became magnified. So 23 hours faster in rapid testing arm in the cephalosporin resistant subgroup, and 29 hours faster in the rapid testing arm in the carbapenem resistant subgroup. And then we wanted to look at appropriateness of antibiotic changes, not just timing of antibiotic changes. So we coded antibiotic utilization from time 0 to 72 hours after randomization, following the ESCMID and the IDSA gram-negative treatment guidelines to determine if something was appropriate treatment or not. And you can see that the different colors reflect no treatment, under treatment, appropriate treatment, or over treatment. And basically, you can see greater movement in the appropriate treatment group in the rapid testing arm versus the standard testing arm within 12 hours or 24 hours. So faster appropriate changes in the rapid testing arm. So in conclusion, rapid testing was not superior to standard testing using the DOOR primary outcome, but we did see that there was faster targeted antibiotic therapy and greater provision and adherence to stewardship when we used rapid phenotypic testing. We did see fewer patients hospitalized at day 30 in the carbapenem resistant subgroup, and there was a trend towards shorter length of stay in subgroups with resistant infections. There are a number of strengths and limitations of this study, which are outlined in the publication, and I'll refer you to that. But I do want to end with just some thoughts about what influences the clinical impact of new blood culture diagnostics. So one of the first things that's important to consider is the breadth of target organisms. If the diagnostic does not cover the vast majority of organisms that cause bacteremia, it will not be so useful. Also, does the diagnostic include target organisms and resistance markers that align with local epidemiology? Of course, that's important. Does the test contain susceptibility results for antibiotics that are available and used in clinical practice? Because if it doesn't, it will be less useful to clinicians. Is the faster turnaround time clinically relevant and sufficiently faster than a standard of care. Otherwise, it will not be considered an improvement and may not necessarily be adopted. Does the workflow of the test and the throughput of the test align with laboratory staffing and specimen volumes that a laboratory sees? And we learned some important things about implementation, that it's important to pair diagnostics with effective therapeutics and stewardship and clinician education to nudge clinicians to act on the results, especially if it's a test that they're unfamiliar with. And lastly, I do want to emphasize that we've learned through the FAST trial and others that we've conducted that there are many measures of clinical impact beyond mortality, which is the holy grail, but really a challenging outcome to impact. So with that, I will end. Back to you, Alec. Thank you for your time.
Thank you, Professor Ritu, for that very insightful talk from the FAST trial. So again, if you have any questions for Professor Ritu, please type them in the Q&A box and we will address them later. Thank you again, Prof. Ritu. Okay. Our next speaker is Dr. Beth Amiot, who serves as one of the Alliance Director for the Infectious Disease Diagnostics portfolio at CARB-X. Her work mainly focuses on advancing innovative technologies addressing antimicrobial resistance from product development to commercialization. Dr. Beth, over to you.
Great. Thank you very much, Alec. Thank you for the invitation this morning. I'm really happy to be here. And I'm happy to give just some insights that I have gained over the years in product development and how you might want to think about your new product as you're developing it in the early phases. And I'll actually overlap on some themes that Dr. Banerjee just highlighted in her conclusions and that I expect that we'll hear in the next presentation as well. So here, as I said, we'll look at some common gaps and oversights that product developers may encounter in their early phase product development. I want to start just by quickly, see if we're advancing here, introducing CARB-X, my association. CARB-X is a global nonprofit partnership that's focused on accelerating research and development for diagnostics, preventatives, and therapeutics that are addressing the rising threat of drug-resistant bacteria. In my role as an alliance director for the diagnostics pillar of CARB-X, we bring into our portfolio and fund diagnostic products that are in their early stage of development. And so this gives me some insights in order to be able to speak to you about things to consider early on your path to product development in order to de-risk your innovation, share some gaps and pitfalls and oversights that we see in product development or that I've experienced in my own life as a product developer, and just quickly touch on what funders and partners in your product development pathway might be looking for that goes beyond technical performance. So I asked Google AI to just help me illustrate some of the commonalities and differences in the mindsets that you might want to have when you're developing a new diagnostic. So on the right, you've got your technology mindset. This is your innovator mindset, where you're really focused on your new discovery, your new idea, your technology, your platform that you're developing, the thing that you're really excited about and that motivated you to try to build a diagnostic. And then you really want to pair that and merge it, meld it with a product mindset. This is where you really need to be thinking about how that technology is going to facilitate this product, which will give you diagnostic clarity, accurate and reliable results that will inform treatment decisions like we just heard about and that deliver value to the healthcare system overall. You really need those elements to be defined in order to make your technology have the impact that you want it to have. And so what are some things that you can do to try to get into that product development mindset to build that fit for purpose product? And I would say you want to be thinking about all of those details and key elements early in your process and revisit them often. You don't have to lock them at the very beginning. You don't have to define every element of your product, but you need to be thinking about that and how that impacts your design and how it will ultimately impact your validation requirements. So really the questions of what, who, where, how is this testing going to happen, and ultimately why. And to what impact. So some of these things that you see listed here are things that you would find in a product instructions for use. You've got your intended purpose of the test, your intended use, your use environment. These are all things that you want to think about early, because you can tailor how you design that product to make it work in those environments where you'll have the biggest impact. And so you want to go through each one of these elements. There are more, but these are a really good start of truly what are you diagnosing? What are you going to measure in order to make that diagnosis? What status of infection are you going to be diagnosing? Who's your patient population? What sample type are you using? Is that the right best fit sample type for your diagnostic? What is the setting in which you want your testing to occur? And then it all goes into how will it get used either in the laboratory or at the point of care, or even for at home or self-testing. So all of those conditions will influence how you design it, what features of your product are most important to bring the best usability and utility. This next, the how and why of your product really get at what Dr. Banerjee was pointing to at the end. What results are you giving? Are they complete? Are they comprehensive and sufficient? Are they easy to interpret? How easy are they to interpret? Will the results get to the person who needs those results in a timely manner? You can have a very fast test, but if you don't have the infrastructure and the system to get the sample to that fast test and then get the results out to the clinician or the decision maker at the end, then it's not really as fast as you've designed it for. So that environment that it's going to be used in is important, and what the results are. How will they be actionable? And are there things that you can design into it that will make them more actionable? And in the middle here, the third bullet point down is, will it change clinical care relative to the current standard of care, which we just heard about? And if it brings value to the health system, how will people implement it? Are there clinical guidelines that exist right now that will allow users to adopt the tests that you've made? If not, what can you do to start to influence clinical guideline development if you're going into a new space, or maybe bringing something where there hasn't been a diagnostic before? The other things on this slide: manufacturability, shelf life, stability, regulatory requirements, cost, market access and demand, willingness to pay. It's a long list. It's a lot to think about, but the earlier you think about these things and start to build your roadmap, the more chance of success you're likely to have. It will also allow you to define that minimal viable product for your use case, which will help you when you come upon hard decisions that you have to make during product development. So a couple of tips that I have that you may want to consider is, early on, draft that preliminary instructions for use, help you define all those items that I just mentioned. Revisit that draft often and adjust as you learn more about your product. This middle column is, go on a visit if you can. Go to the places where you want your test to be used. See what the environment is like. See what the conditions are like. A lot of times we have assumptions about who will use the test, what their day looks like, how they're going to implement it, and we don't always get it right. So whenever you can, go and see it in person. Look for the pain points that you might be able to design mitigations for. Understand what happens if you have a failed test. What impact does that have on the workflow and the people running the test, or the impact on the patient for those delayed results? And then the final column here is, if you can, prepare some test patient scenarios, distribute those to clinicians, understand how will they use that test result. When is it likely to lead to an action? When is it likely to be misinterpreted? When is it likely to reinforce what they might already be doing? And then after you've learned all of that, really try to identify the minimum performance targets that you need that match to that intended use and actionability of the test. So once you've defined all those requirements and you start working on your development, there are some other things experimentally that that we sometimes see or that I've sometimes experienced in the development process. And I'm just going to focus on the ones here with orange. It's a big list. It's a lot to think about. But if we go from the upper left and across the top and back around, especially because we're talking about bacterial diagnostics and resistance, contamination and the risk of contamination and things that you can design to prevent it is really important. Using clinical samples early in your product development process, there's a lot of variability and potential interference that comes from the samples, and there's a lot of logistics to getting the right access and the right quality samples for your development process. Really thinking about sample and reagent stability while you're doing your development, because this can impact your experiment and the interpretability of your experiment, and is something that you want to find out early in order to mitigate. Another recommendation I would have is implement sample and process controls early in your product development. This will help in your experiments and your interpretation of your experiments throughout the design process. Similarly, down in the lower left, having consistent protocols and acceptance criteria while you're doing experiments so that you can interpret as you progress through your optimization and development process. And looking for silos or mismatches between your different groups that are building your product. If you have reagents, if you are building an instrument, if you're building software, make sure those teams are working together, not only for the details of the work, but also their timelines and their dependencies upon each other in order to make those timelines line up with each other. And finally, I'll touch on the last topic, which is here's your technology, here's your product vision. What else might funders or partners that you might want to pair up with during product development be looking for when they're looking at you as a product developer that go beyond that technical performance? And a big one I would say is your company structure and culture. Do you have the expertise and experience? Have you taken a product to market before? Or do you have somebody on your team that has? Clarity and robustness of data and communication of your data. That's really important. Openness to collaboration and input. Showing your resilience and your success in identifying and overcoming challenges that you've had in your product development process, and that you have clear and strong decision-making processes. Setting those requirements early will help in that decision-making. Other considerations are, do you have clinical partners set up? Are you thinking about pilot studies? Do you have plans for generating evidence about clinical utility? Realistic planning and prioritization, realistic scopes, minimum viable product definition, time, budget, and resources. And can you demonstrate progress on your manufacturing? Have you thought about your quality management system and risk assessment processes and your regulatory needs? All of those show that you're on your path to going from early development to early phases to a successful product development and commercialization. Thank you.
Right. Thank you, Dr. Beth, for your valuable insights and perspectives on product development. So again, if you have any questions for Dr. Beth, please type them in the Q&A and we will address them later. Okay, moving on to our next speaker, I would like to introduce Dr. Wang Reifen. Dr. Wang is the Chief Executive Officer of Diagnostics Development Hub, or DXD Hub, Singapore's national platform for diagnostics development, hosted at Singapore's Agency for Science, Technology, and Research, or ASTAR. Dr. Wang also leads Diagnostics Cooperative under PREPARE, a dedicated national pandemic research program hosted by the Communicable Disease Agency under Singapore Ministry of Health. Dr. Reifen, over to you.
Thank you very much for the very kind introduction and I think the invitation to share at the webinar today. So really the key questions that we would like to address in this segment are around whether or not there is a common regulatory pathway for Asia market access, similar to the CE marking for European market access, and whether manufacturers could submit the same technical dossiers from CE or other regulatory bodies for pre-market review in this region. So unfortunately, the quick answers to both questions are no. There is currently no harmonized regulatory framework in Asia. This means that all devices, including in vitro diagnostics, need to be registered with the respective national regulatory authority in the country of interest, and that also means that manufacturers need to consider the requirements of each country individually for product registration and market access. So I think in the next 10 minutes or so, I hope to maybe unpack some of these country or region-specific regulatory nuances by going through a very quick overview of the regulatory landscape across Asia, but with a focus and emphasis on a couple of low middle income countries which are of priority to the current Detect Asia program. And I think we will start by looking at the landscapes in Southeast Asia, or otherwise known as ASEAN. And then we will travel south to South Asia to look at countries such as Nepal, India, and Sri Lanka. Oops. So I think fortunately, it's not all too depressing for all of us developing innovative products. I think within Southeast Asia, there has been an ongoing harmonization regulatory efforts through what we call the ASEAN Medical Device Directive, or otherwise known as the AMDD in short. Briefly, AMDD is an agreement that has been signed in 2015 by all ASEAN member countries. And this was an effort to really facilitate trade and market access for medical devices in ASEAN by really reducing the technical barriers that is required for product registration in the region. So under the AMDD, while all devices still need to be registered with the respective national regulatory authority in each ASEAN country, the AMDD really sets out a common guidelines for product risk classification across the region, while it also provides a set of common technical and compliance requirements that are required for both pre-market product review as well as post-market surveillance and reporting. And I think a key component of the entire AMDD framework that is really useful to many of the device manufacturer is the Common Submission Dossier Template, or CSDT in short. So the CSDT So the CSDT really is a set of standardized and common formats for technical documentation for device registration that include IVD in the ASEAN region. So the use of the CSDT really helps reduce the time that is required for manufacturers to prepare the regulatory technical dossiers for pre-market reviews for submissions to countries in the region. I think for manufacturers that are very experienced in other global markets, they will find it relatively straightforward to work with the CSDT because the core elements of CSDT really are built based on global standards such as the IMGRF. Essentially, what this means is that manufacturers that are already exporting to Europe or other regulated markets could adapt or adopt and adapt its existing technical reports and other technical documents for market review in the ASEAN region. However, given the diversity of countries in this region, there are a couple of country-specific requirements which I think would be helpful for manufacturers to be aware of. And some of these are actually listed on the slides here. First of all, I think while CSTT submissions are almost universally accepted in English, it is also a very frequent market approval requirement to have both product labeling as well as product IFU translated into national language of the ASEAN country of interest. And second of all, I think while most ASEAN countries accept international performance data for many IVDs, local performance data is sometimes required. I think especially so for products that are considered high risk, novel, or intended for infectious disease management, or intended for self-testings or near patient testings. I think the major exception is the Philippines, which actually requires in-country testing data by its designated labs for most IVD products. And another key stakeholders for market registration in the region really is this concept of the local authorized representative. The local rep is actually required by all ASEAN countries for pre-market registration as well as imports and post-market surveillance. While most countries accept multiple local reps, in Indonesia, which is one of the country of interest, actually only one local rep is allowed for any product at any point in time. What that means is that if you want to change your local rep, you will have to cancel the current registration with the Indonesian regulator and take the product off market while the new local rep submits the application for regulatory review. Is a common struggle for a lot of countries that are new to Indonesia markets. So I think what the AMDD, even though it has been established since 2015, each ASEAN member states could actually choose when they would fully implement the AMDD. And the table on this slide really shows the current implementation status of AMDD in the various countries. I think of interest to the program today, Indonesia is in fact among the countries that have more or less fully implemented AMDD, while the Philippines have adopted CSTT for general medical devices, and the requirements for IVD as well as the adoptions of the full AMDD framework is an ongoing effort. Now, I think we will switch gear to have a quick look at the key regulatory framework in countries like Nepal, India, and Sri Lanka. Unfortunately, at the moment, I think the landscape here is a bit more heterogeneous. There isn't a sort of a regional regulatory harmonization effort ongoing. While countries like India and Sri Lanka have a risk classification system that is rather similar to major regulators globally. In Nepal, there is currently no official product risk classification. So I think a market access tool that is very useful to many device manufacturer is the concept of regulatory reliance, where a device which has been pre-registered with a recognized regulatory body will receive an expedited market registration in the country that has officially endorsed the specific reliance program. The slides here really shows sort of a brief summary on the adoptions of regulatory reliance as a concept among the key countries of interest to the program. For example, in India, all the IVD products that have been registered in markets such as Australia, Canada, Japan, the EU, USA, and the UK are eligible for Reliance program. And what this means is that if a product have been pre-registered in those markets, and the product also have a predicate device in India, in-country clinical performance evaluation will be waived for the products. Manufacturer can submit directly with the data from from overseas. But it's important, I think, helpful to also note that in the event that there is no predicate in the Indian markets, even if a product has been pre-registered in the Reliance markets, the Indian authorities still will require in-country clinical performance evaluation for class B, C, and D IVD products. On the other hand, I think in Sri Lanka, the local product testing is entirely waived for products that fall under the recognized Reliance program listed on this slide. And unfortunately, in other countries of interest, including Nepal, Indonesia, and the Philippines, there is currently no official regulatory Reliance program for IVD products. And I think a common sort of mistake that we have observed in the last 12 years or so is that I think some of the speakers earlier on has alluded to this as well, is that many manufacturers, I think especially small medium enterprises, tend to consider regulatory and quality requirements pretty late in the product development cycle. And what this often results in is very costly rewards and rethinking of market entry strategy, and therefore, significant delay in market entry. At DXT, what we typically work with is a set of guidelines where we tend to define product intended use and product claims very early in the development to what Dr. Beth mentioned earlier on. We'll consider the local regulatory requirement at the get-go of the product development journey. Well, especially in low middle income countries where the regulatory science is still evolving, requirements could also evolve. It's also very critical to engage very early with the local representative as well as the local regulatory authority to stay very updated with the most recent and relevant product-specific requirements. And as part of our regulatory strategy for market access, we leverage a Reliance program as much as possible to speed up multi-country market access. I think last but certainly the most importantly, it is very critical to really invest in building a sustainable local partnership, either through product co-development with local key KOLs to better define product market fit, or to explore some of the local regional manufacturing support to leverage local government support, or to build really what we call global diversification in supply chain. And I think I would end the sharing with a very brief introduction of DXT Hub, the organization that I work for in Singapore. So DXT is a Singapore government-funded platform, and we focus on building diagnostic products through close partnership with both the public and private ecosystems. We are essentially a hands-on product co-development partner where our team of 70 plus development scientists, engineers, quality and regulatory experts run a set of ISO 13485 compliant laboratories that support end-to-end product development, starting with the definition of a fit for purpose, target product profile, identification of a regulatory strategy for market access, followed by what we call hands-on technical product optimization and regulatory verification and clinical validations, really gearing towards regulatory clearance in Singapore and in many other countries that are relevant to our industry partners. And I think on the right here are some of the companies that DXD has partnered with in the past to bring products from the bench to various markets in the last decade or so. So with this, I will end my sharing today, and we look forward to working with many of you on the Detect Asia program. Thank you.
Thank you so much, Dr. Ruifen, for sharing your expertise and perspectives on the regulatory challenges across Asia. Again, if you have any questions for Dr. Ruifen, please type them into the Q&A box below, and we will address them during the discussion session later. Allow me now to introduce myself. My name is Luca. I'm an infectious disease physician and one of the project leads for Detect. I will be moderating the second part of today's webinars, during which we will share details about the upcoming open call. For this session, it is my pleasure to introduce Prof. Moin. Prof. Moin is an infectious disease physician and currently serves as co-director for Advance ID, a large clinical trial network based at the National University of Singapore. She has extensive experience leading large multi-center clinical trials and coordinating research networks across Asia and globally. So, Moin, over to you.
Thanks a lot, Luca. Hello, everyone. Thank you for attending our open call webinar. Okay, let me start off with giving an introduction about our DETECT program. So DETECT program is one of the anchoring pillars in Advanced ID, focusing on diagnostics, looking mainly at antimicrobial resistance, fungi, as well as bacterial infections. This funding was given to us by the Novo Nordisk as well as the Temasek Foundation last year, really focusing on improving the laboratory capacity across the region. Sorry about the transition. Okay, so as an overview of the whole DETECT program, as I've mentioned, the DETECT Asia program is one of the anchoring pillars, alongside with our other clinical trials and platforms targeting therapeutics as well as prevention and surveillance, again, mainly looking at antimicrobial resistance infections in the region, especially the severe infections such as bloodstream infections and ventilator-associated pneumonia. And for this DETECT program, we have four main work streams. The first work stream that we have recently completed, or actually still ongoing in some countries, is to evaluate what's the baseline capabilities of laboratories in countries across Asia and especially LMICs in other continents as well. So we have developed a standardization tool together with our partners such as GAPI, WHO, CABEX, and a lot of the investigators, especially in LMICs, and I'll be able to share the results with you in the following slides. Next, after getting to know these sort of baseline capabilities in the labs across the region, we are currently talking to many labs in our partnering countries, especially focusing in Nepal, the Philippines, and Indonesia on devising locally driven solutions. And a lot of these solutions are looking at implementation science and identifying low-hanging fruits on how to optimize work processes within the lab and as well as communications between the lab and practicing physicians. So as we all know, rapid diagnostics cannot be just plugged into any lab. There has to be efficient workflow and some basic both hardware and software for us to operate the diagnostics well. So therefore, the second work stream links directly to the rapid diagnostic work stream, the third one and the focus of today, which is to assimilate rapid diagnostics into routine care. And because we do have a ongoing surveillance study across 19 countries and many hospitals, especially in LMICs, we will be able to make use of the surveillance studies as well as the dashboard, as you can see on the screen here, to detect the impact of our diagnostic work across our study sites in these countries. So let me share with you the global survey results. We have started this exercise about a couple of months ago, and so far we've already received over 500 responses from 47 countries. This is no easy feat for the team here because, as you know, all of us are really, really busy in our labs, and the survey, even though we try to keep it concise, is still fairly extensive. So today we are mainly focusing on the TPP of the diagnostic test we are looking for. So out of all the TPP questions, we have identified these four main features that the investigators or the practitioners would like to focus on, or the disease burden that is really visible in their own setting. Firstly, the attention in terms of the syndromes is in bloodstream infections, meningitis, encephalitis, as well as in lower respiratory tract infections. And as we know, these are the infections that really cause very high mortality in the region. And secondly, there's a lot of interest in looking at gram-negative bacteria as well, and this also happened to be the WHO priority pathogens in terms of AMR, antimicrobial resistance. And the top concern is Acinetobacter baumannii, and that is also from our ACON HAI or surveillance study has been identified to carry the highest mortality burden in the region. And this is followed by Klebsiella pneumonia as well as E. coli. In terms of the resistance genes, it's no surprise to see that carbapenemase are the top priority in LMICs, followed by extended spectrum beta-lactamases and the other genes associated with MRSA or VRE. And this is really because carbapenem resistance and gram-negatives they do have a lot of mortality burden as well as quality of life and extending our hospitalization in the region as well. So last graph over here, plot here shows the desired features. So the main wish from the investigators or practitioners would be that they would like a true point of care test. And what we mean by true point of care testing here is that there's no need for a machine or electricity or sophisticated consumables for these tests to be carried out. An example of a true point of care testing would be like a COVID swab test that we are very used to. Secondly, would be rapid testing. So in terms of the time from sample collection to reporting being done, that has to be as short as possible. And lastly, or thirdly, most important, we are looking at affordability. And in the tables following this slide, we'll be sharing with you the price points we are focusing on. So aside from these four main questions, we also did a very simple clustering analysis of the responses in the surveys, and we noticed there were two main patterns. Firstly, they are mainly responses from the low middle income setting, and the preference is for a very affordable point of care test. And the testing should be able to be done near the patient or at bedside. And the second group of testing is that the practitioners would prefer more advanced testing, both providing the identity of the pathogens as well as the resistance genes or phenotypes. And in this group of practitioners, they value functionality over potentially affordability, and they prioritize antibiotic susceptibility testing profiling. So we'll be publishing this table shortly following this webinar. These describe the exact target product profiles that we are looking at. So you can see here the first column refers to the characteristics in terms of intended use, clinical specimens that we are looking at, reflecting the syndromes that we are the most concerned about, and followed by the minimum requirement as well as the optimal requirements. So coming back to the TPPs and also the price points we are looking at the sample capacity of at least 5 to 10 samples per run, and ideally point of care testing and fully automated or semi-automated, as well as minimal training requirements. And the price point ideally should be less than 15 to 20 US dollars per test, and time to result is between 1 hour to 1.5 hours. So in terms of evaluation of the technologies, what we are looking at, again, the form will be circulated after this webinar on our website, as well as through email. We'll be using a semi-quantitative questionnaire with a Likert scale, is 5 points, and each application will be carefully evaluated by 3 members from our technical evaluation committee. And our technical evaluation committee is formed by regional experts from all the major countries within Asia. And the domains we are looking for is very closely aligned with our TPPs. Namely, they are the clinical and stewardship impact, technical feasibility, implementation and sustainability, and lastly, scalability and affordability. And the timeline-wise, we are launching the open call on the 6th of July, and that will last about 6 weeks. And the evaluation will end about mid-September, and all the applicants will be notified of the outcomes in end September. And for the shortlisted technologies, we will be calling you for a pitch to the TEC. So what's in for the shortlisted technologies? We'll be finalizing 2 to 3 of these. And what we can offer for these technologies is a joint effort from DSD Hub as well as Advanced ID. Firstly, at DSD Hub, we'll be able to offer wet lab evaluation. So we do have a very wide collection of isolates from especially LMICs in Asia. We can do that in the lab setting by DSD Hub in Singapore. And in terms of clinical evaluation, we can conduct that in our study sites across Asia, comparing to standard of care testing, and we'll be also able to provide health economic outcomes, clinical outcomes, on top of just process measure or diagnostic turnaround time outcomes. Yes, I think that's the last slide. I would like to thank our project leads here who's been working very hard to make this possible, as well as Ritu, Beth, and Rayfern for your time to give us your insightful teachings. Thank you, everybody.
Okay, thank you, Prof. Moin, for sharing the results and going through the TPP. My name is Cleven, and I'm one of the project lead for the DTEC program. We have some questions in the Q&A box. We still have some time, so we can go through that. The first question is by Joanne from Loop DX. Are there any ASEAN countries that recognize each other's registrations? I heard that Thailand recognized HSA approval without needing to resubmit for some countries. If not, does approval in any of the ASEAN countries grant an expedited or abridged pathway in other ASEAN countries? Maybe Dr. Rayfern, would you like to take this question?
Sure. So I think as the participants share, Singapore actually had a pretty established regulatory reliance program with major markets in Europe, in the US, as well as within ASEAN. We have between HSA and the Thai FDA, there is established regulatory reliance program. Similarly, I think recently last year, there was an announcement of a regulatory pilot between Singapore and Malaysia. So with the reliance program, what this means is if a product registration carries the exact same intended use, and that has been registered in Singapore, there will be an expedited review by the Thai FDA and vice versa. So these are mutual reliance program. There are also other programs that the Singapore HSA is, I think, working very hard to establish, and we look forward to the official announcement soon to come.
Thank you, Dr. Redfern. The next question by Garrett. What incentivized the Indonesian policy requiring diagnostics providers to withdraw registration and remove their product from the market when switching local reps? Maybe Dr. Redfern as well?
I think every country has its own jurisdiction when it comes to regulatory requirement, considering the local context in business and so on. So I think this is a key area that I think we pay attention to when it comes to bringing solution to Indonesia. I think there are sort of efforts to look at who is the best or most appropriate local partner. And then I think for more experienced manufacturers, there are also opportunity to also consider setting out a local incorporation to register the product under the company directly. So I think there are, it's like any countries will have its own sort of sets of consideration when it comes to regulatory requirement. Thank you.
Thank you, Dr. Redfern. We have one more question from Jose. I'd like to know if diagnostics in the areas of phage therapy for targeting AMR will be eligible for the announced call. Maybe Prof. Moin, would you like to take this question?
Thanks, Cleven. As we've shared in our TPP, we are mainly focusing on identifying the bacterial pathogen, as well as antibiotic susceptibility and its associated genes in the TPP. In applying phage therapy in the region, and especially in LMIC settings, it's being currently offered at a compassionate basis in the absence of very robust regulatory science around it, and also lack of strong clinical evidence. Yeah, so therefore it is not a priority in the current call. Yeah. But just maybe of interest that we are actively in Advanced ID looking at phage therapy. We've collected bacterial samples from the region and developing a regional-wide phage, and working on this area to develop either randomized controlled trial evidence in individual patients as well as in environmental clinic.
Thank you, Prof. Moin. Maybe due to the interest of time, we'll just take one last question. So we have from the anonymous attendee, for the application, all criteria for the TPP is very focused on pathogen identification and resistance testing. However, in our case, our test is focused on assessing which patients need or don't need antibiotic therapy, not as much as identifying pathogens and resistance. Will this still fall within the scope of the program? Prof. Moin, would you like to take this?
It's a really important question to answer to, for example, differentiate viral versus bacterial infection, or differentiating contaminants with true pathogens. However, in our survey, this did not come up to be one of the focus areas, and therefore it's not prioritized in the current open call. I wonder if Beth, I understand CARB-X is also working on a lot of diagnostics. Would you have any advice on other calls, opportunities?
I would think that, be aware of those funding entities and their schedules. So CARB-X has open calls twice a year, typically on the same theme in the spring and the fall. This year we're focused on diagnostics for neonatal sepsis, which it does have a broad indication. It is not specifically targeted at specific pathogen identification. So you could look at the minimal entry criteria to see if that's applicable. It is a very specific patient population. So it would depend on if that fits with what your technology is. But this is a growing area of investigation and development. And I think keep your eyes open for other opportunities. I think they'll be out there.
Thank you, Prof. Moin and Dr. Beth. Due to the interest of time, if there are still any further questions, there is the QR code here. So you can contact the project team or via the inquiry form by scanning the QR code here or via the link, or you can email any of the project leads and we will try to answer these questions as soon as possible. Thank you so much.
Thanks, everyone.
Thanks,
Thank you so
everyone.
much.
Thank you.
Thank you.